Theistic Evolution Critique, Population Genetics. We've been looking at the book Theistic Evolution, a scientific, philosophical, and theological critique. And the book deals with one particular theory of how reality is put together and came to be. There are several theories. You can have what I would call a young life creation, various flavors of that. Then we have what is traditionally known as old earth creation, but which should probably be known as old life creation, because those tend to hang together. And then there is with theistic evolution that is friendly to intelligent design. That is, it came about by some kind of a theistic evolution process, but you can tell that it's designed. And then there's what I would call non-intelligent design theistic evolution. It looks for all the world like atheism, but God really did it that way. And then finally there is flat-out atheistic evolution. Now the book is not actually aimed at atheistic evolution, which is where you would normally assume. This is actually aimed at non-ID theistic evolution. The chapter that we're going to look at is by Ola Hössjer, or Hössjer, I'm not sure how you pronounce that, Hössjer and Colin Reeves. The same three authors that were for the chapter before, in slightly different order. And this is in part one of the book. It's the scientific critique of theistic evolution. So we're dealing with science here. The case against universal common descent and for a unique human origin. This is part two. And finally, the chapter itself is entitled An Alternative Population Genetics Model, which is where I got population genetics in the title on the top of the screen. The summary starts out, what can be said about human history from DNA variations among us today? Population genetics is used in academia to infer that we share a common ancestry with apes, that most of our human ancestors emigrated from Africa 50,000 years ago, that's of course more or less, that they possibly had some mixing with Neanderthals, Denisovans, and other archaic populations. In fact, I would say certainly at this point, and that the early Homo population was never smaller than a few thousand individuals, which of course destroys the idea of an original couple. It uses mathematical principles for how the genetic composition of a population change over time through mutation, natural selection, genetic drift, and other forces of change. In this chapter, we investigate the assumptions about this theory and conclude that it is full of gaps and weaknesses. We argue that a unique origin model where humanity arose from one single couple, we'll look at that note in just a minute, seems to explain data at least as well, if not better. We will finally propose an alternative simulation approach that could be used in order to validate such a model. The note says the use of the term first couple will undoubtedly raise the issue of Adam and Eve in the reader's mind. We, the authors, each have our own views of the reading of Genesis, which presumably means that they have different views from each other as well. Our goal here is to show that the argument against a historical Adam and Eve made by some scientists is not justified by the scientific evidence, and that there is a real possibility of a founding first pair. And that's really what we're going to be looking at. Okay. The chapter begins, we all have a genetic fingerprint. The more closely related we are, the more similar our fingerprints. Genetic data can be used for a number of purposes: to find out whether we carry a gene of an inheritable disease, to ascertain that a young man is the father of a newborn child, to find evidence against a suspect of a crime, or to retrieve our ethnic mix at Ancestry.com. In this chapter, we will investigate what human genetic data has to say about common ancestry? In academia, the prevailing view is that human beings are descended of ape-like ancestors whose numbers were never less than a few thousand individuals at any time in history. In other words, there was no Adam and Eve, at least as a single pair. There's another possible scenario, though, that of an original first pair. This scenario has never been adequately tested scientifically using the methods of population genetics. Given the importance of this subject, it is imperative to rigorously examine both scenarios. In this chapter, we describe a method to test them against each other. Population genetics. Population genetics is a discipline that describes how the genetic makeup of a group of people changes over time. It has numerous applications, but here we will use it as a tool for comparing different scenarios of human history. Before doing so, we will first review some basic principles of genetics. Genetic information is stored in all our cells as 46 chromosomes, 23 of which are inherited from our father and 23 from our mother. 44 of these are non-sex or autosomal chromosomes and come in almost identical, or what they call homologous pairs. The remaining 2 chromosomes determine our sex. Females have 2 copies of an X chromosome, one from each parent, whereas males have 1 Y chromosome from the father and 1 X chromosome from the mother. There's also genetic information in the mitochondria. Those are organelles in our cells, inherited from our mothers. Well, mostly. Almost all. The 46 chromosomes of the cell nucleus and the mitochondria of the cell cytoplasm are molecules. The genetic information is stored in these molecules as nucleotides of A, C, T, and G, linked together to form the double-stranded DNA helix. Each nucleotide or base pairs with another in a stereotypical fashion, A with T and C with G, and I think most of you are familiar with that. And here's a single nucleotide that has been changed, and of course it changes the opposite one as well. It's usually counted for the coding strand. Most parts of DNA are the same for all humans, but those that vary are called, polymorphisms. They are the ones that make us genetically unique. A single nucleotide polymorphism is the most common kind of variation. For each base pair of the DNA molecule, the convention is to refer to only one of its 2 nucleotides, the one located on the coding strand. Although what do you do if they're both non-coding? That makes it interesting. This nucleotide usually exists in 2 variants at an SNP, for example, C or T, also referred to as the 2 alleles of the SNP. A typical genome differs from the human reference sequence, which has the most common SNP at all positions, at about 4 to 5 million nucleotides, slightly more than 0.1% of the human genome. So we're like 99.9% identical to each other, and a little more if we're more closely related. Since our genomes are not identical, population genetics can tell us something about human variation in history. We can study how big the genetic differences in worldwide human population are, and indirectly how these differences have changed in the past. We can also compare the genetic makeup of individuals from different regions like Europe, Africa, the Middle East, or East Asia, or study smaller groups of people like the inhabitants of Sardinia, Iceland, or Polynesia. Population genetics is a discipline that uses mathematical methods to quantify how genetic differences vary among individuals between geographic regions and over time. Starting in the 1920s, much of its theory was built by prominent mathematicians and geneticists like Ronald Fisher, Sewell Wright, John Haldane, Muthu Kimura, and Samuel Carlin. Some of those names are probably familiar to you, including Fisher's t-test. Its theory is built on the principle of small stepwise changes, and as such is a good tool for describing microevolution within species, which of course humans are, and some limited degree of speciation. This has many applications, for instance, animal and plant breeding, and also wildlife management and conservation biology, where the viability of species and their adaptation to environmental changes and inbreeding can be quantified and estimated over time. However, population genetics was also intended as a method to explain how small changes could lead to big ones. In other words, it was largely founded to support macroevolution and common descent, based on the idea that macroevolution was merely microevolution writ large. But today, It is possible to use it for the opposite purpose, to show how unlikely macroevolution is. In the following sections, we will describe this in more detail. The mechanisms of population change. Our genomes are scrambled images of our ancestors' genomes. In order to understand how history has scrambled our ancestral DNA, we first need to describe mechanisms that cause the genetic composition of a population to change over time. These mechanisms reflect population behavior, or demography, as well as genetic inheritance, and thus can tell us something about a population's history. They can be summarized as follows. Mutations are changes to DNA. These mutations are typically copying errors. The ones of interest in population genetics are those which occur when germ cells are formed during a special type of cell division called meiosis, which halves the genetic material of ordinary cells and reduces the number of chromosomes from 46 to 23. These mutations are important since they spread to all cells of the offspring after fertilization. Suppose, for instance, that a single nucleotide of a non-sex chromosome is changed from A to a G in a sperm cell, which fertilizes an egg. The probability of mutation occurring is very small, about 10 to the minus 8 per nucleotide per generation in nuclear DNA, but you have like 3 times 10 to the 9th nucleotides, so that means that there's probably 30 of them in the entire genome. As the resulting embryo develops, all the cells inherit that mutated allele G from the father, as well as the mother's version, say, A. The two versions of the inherited DNA form a pair, AG, of alleles. Could be A, could be G, in the new individual. That individual will in turn pass on either the A or the G to each of its offspring, at random usually. Mutations also occur in sex chromosomes and mitochondrial DNA. Genetic drift means that alleles will change in their frequency from one generation to the next simply by chance. Most of you are familiar with that. Natural selection is similar to genetic drift. Similar, but not identical. Some parents have higher fitness and produce more offspring than others due to differences in their ability to have children or differences in the survival of their children to the next generation. If parents with more copies of an allele, say A, at an SNP tend to be more fit than those that have more copies of the other allele, say T, the frequency of A will gradually increase in the next generation, and eventually T may disappear in the whole population. On the other hand, if individuals with one copy each of A and T, allele pair ALT, are more fit, because they have some advantages there, the frequency of both alleles will tend to stabilize over time. That apparently is true in some HLA antigens. Recombination is a way of generating more diversity between chromosomes by shuffling the arrangement of alleles along the chromosomes. No novel DNA is produced, but existing DNA is combined in new ways. And this is an illustration where A was originally paired with C in this particular one, T was originally paired with G, and then you cross over, and now the A is paired with the G that was down here, and the T is paired with the C that was up above. Recombinations make variation at different parts of the chromosome more independent, as they gradually break up association between alleles. This happens, for example, in Figure 16.2, which we just saw. Since recombinations occur more often if the SNPs are far apart, the association between a distant SNP pair will be smaller. There's more opportunity to do crossover between it. Colonization, isolation, and migration. Especially in the past, humans have been more or less isolated by distance, with mating couples living nearby. Sometimes new subpopulations are formed, colonization, and occasionally men or women migrate over longer distances to find their spouses. It is evident that migration will decrease the differences between subpopulation, whereas isolation will increase them. Statistics describing genetic variation in a population. There are certain statistics that describe the current genetic variation of the human population. Nucleotide or allelic diversity roughly says how many SNPs there are in the overall population. Allele frequency spectra show how many rare and common alleles there are in the population. Linkage disequilibrium plots give a description of how recombination has affected the variation we see along chromosomes today. The more recombination events there were in the past, the shorter are the chunks of DNA that have been inherited together. There are also other statistics that can be used to describe the degree of differentiation between subpopulations. When population geneticists try to reconstruct human history from genetic data, they combine different demographic scenarios, population size changes, colonization, and migration, for example, with the genetic mechanisms of mutations, recombinations, genetic drift, and selection, to see how well these scenarios reconstruct the statistics mentioned above that we can actually observe. There are highly sophisticated mathematical methods for doing this, but it is still very difficult to reconstruct human history. The main reason for this is the lack of data from the past. It is indeed possible to sequence DNA from ancient people such as Neanderthals, and in recent years this line of research has exploded. In spite of these advances, ancient DNA is still so sparse that, to a large extent, genetic analysis of human history will continue to rely on DNA samples from the most recent generations. With little historical data, any reconstructed genealogy is an estimate only, with assumptions embedded in it. In order to illustrate how the past has shaped the genetic variation of today, let us consider allelic diversity. It is a balance between genetic drift, mutations, recombinations, selection, and migration. Genetic drift will, first of all, decrease diversity, since some alleles are going to be lost by chance. And this happens much more quickly the smaller population is. Mutations and recombinations, on the other hand, will increase diversity, since either new alleles enter the population or existing ones are combined in new ways. Natural selection may either increase or decrease allelic diversity, although it usually will decrease it. Diversity will decrease if alleles with higher fitness take over, or if deleterious alleles are lost, as you're getting rid of one kind, whereas diversity increases if coexisting alleles have a selective advantage. That's that AT thing we talked about earlier. Migration and colonization shuffles around existing alleles between subpopulations. The more isolated subpopulations are, and the less migration there is between them, the larger is the diversity in the whole population, since different alleles tend to take over in different subpopulations, whereas the diversity in each individual subpopulation decreases due to isolation and genetic drift. Alternate human histories. There are two important, controversial, and interwoven aspects of human history: the age of our initial population and its size. Age. How can we use allelic diversity to say something about age? Because of the balance between genetic drift on the one hand and mutations and recombinations on the other, population geneticists argue that the younger a population is, or the more recently in its past its size was drastically reduced, a so-called bottleneck, the less variation we see among the members of the population alive today, since there has been less time for mutations to produce new variants. A small amount of diversity, therefore, indicates a young population or an old population that recently went through a severe bottleneck. Can't tell the difference between those two. More refined conclusions can be drawn if we also take the other statistics, allele frequency spectra, LD plots, and subpopulation differentiation into account. For instance, the LD plot of an older population reveals that shorter segments of DNA have been inherited together, since there's been more time for recombinations to shuffle around chromosome segments. Size. Based on the genetic diversity and these other statistics, population geneticists estimate that our population never contained less than several thousand individuals to account for current diversity, and that it probably contained about 10,000 at the time our last common ancestor with chimps, which they estimate to be about 6 million years ago. The rationale of this argument is that the diversity we see in the human DNA today is not only too large to be explained by a single founding, not only too large to be explained by a single founding couple, it is also too large to be explained by an old population that more recently went through a bottleneck much smaller than a few thousand individuals. Too much diversity. It is possible, however, and this is italicized in the book, that a first founding couple were created with initial diversity, each individual with 22 pairs of autosomal chromosomes, and either an XX for the female or an XY for the male. Thus, there would have been 4 total copies of each non-sex chromosome, 2 from each. These 4 copies could each have been created unique, giving rise to a very large number of SNPs, all of which were present in the first generation. This could be true for all of the 22 sets of 4 homologous chromosomes. In the same way, the 3 X chromosome copies, 2 for the female and 1 for the male, could have been created with diversity. Some diversity from the first generation is also possible for mitochondrial DNA. Since the female carries hundreds of mitochondria that could have been diverse, she may have passed some of that diversity on to her daughters. But notice, the male doesn't have that diversity. That'll be important later. So we now have 2 competing hypotheses: common descent of humans with other species, or a unique origin for a first founding couple with created diversity. In the next section, we will juxtapose these 2 models of human history: the standard model that assumes common descent and the other model that assumes we came from the first 2 parents, or unique origin. The 2 models have different starting assumptions, but following the same rules of inheritance and population, but follow the same rules in population genetics in their working out. The competing models, the common descent model. The most widely accepted common descent theory of humanity currently holds that our ancestors diverged from chimps about 6 million years ago. Then a hominid species, Homo erectus, evolved in Africa. It spread to Europe and Asia about 2 million years ago. So we've got all of this stuff going on 2 million years ago. Various archaic species are believed to evolve from Homo erectus over the past 50 to 80, 500 to 800,000 years, including Neanderthals in Europe and Denisovans in Asia. See Chapter 14 for more on this subject. There are 2 main variants within this framework for how Homo sapiens, our species, came about. The out-of-Africa replacement model. According to this theory, modern humans evolved from Homo erectus in Africa 100 to 200,000 years ago. Then they went through a bottleneck that reduced the population size to an order of 10,000 individuals or smaller. A large part of this group emigrated from Africa about 50,000 years ago to the Middle East, Europe, East Asia, and America, gradually replacing existing archaic species. After leaving Africa, all non-African populations have experienced much more recent and long-lasting bottlenecks before they started to grow, which is interesting hypothesis. The second one that's common is the multi-regional evolution, which posits that our ancestors evolved in parallel from archaic species in several parts of the world, possibly with an African dominance. As a consequence, we have to trace human lineages up to 2 million years before they all end up in Africa. The replacement model has been the most popular common descent model of human history for several decades, but there is no distinct boundary between it and multi-regional evolution. An out-of-Africa scenario with some interbreeding with archaic populations is not too different from a multi-regional model with an African dominance. All human populations except sub-Saharan Africa's have about 1 to 2% Neanderthal DNA. In fact, it seems that as much as 40% of Neanderthal DNA is found in at least some individuals alive today. Presumably those are paleoanthropologists who are being criticized by other paleoanthropologists. The unique origin model. By a unique origin model, we mean one in which humanity originates from a single couple. There is still the question of geographical origin. There are at least 2 versions of the unique origin model with different geographical ancestry. African ancestry. This is a scenario by which the first couple lived in Africa. It has many similarities to the out-of-Africa model, except for the unique origin assumption. The subsequent migration scenario out of Africa could be similar for both models. In the next section, we will argue that a unique origin with an African ancestry typically gives old estimates for the age of humanity. How old, you may want to pay attention. Middle East ancestry posits that the most recent common ancestors of all humans lived in the Middle East. The subsequent migration from the Middle East to Europe, Asia, America, and Oceania could be similar to the out of Africa and African unique origin models discussed earlier, in that the migration is the same general direction. However, this model differs from the African ancestry model and out of Africa models by proposing that Africa was colonized from the Middle East rather than the opposite. In the next section, we will argue that in light of genetics, this gives a much younger age for humanity. Comparison of the models. The crucial question is, which of the different scenarios of common descent or unique origin in Section 5 the actual genetic data supports the most? We will consider the evidence in several parts. Differences with other species. A major drawback of the common descent models, both of them, is the difficulty with handling the significant genetic differences between humans and other species. This is treated in detail in Chapter 14, which we've heard already. Variability in human genetic data. The main argument against a unique origin is that the nucleotide diversity within the human population seems too high to make a single founding couple possible. But this is not a problem for non-sex and X chromosome DNA if there was initial created diversity. Although it is not possible to invoke created diversity for Y chromosomes, so you'd expect the Y chromosomes to be more precise, recent studies have shown that its diversity, that is the Y chromosome diversity, is in fact a lot smaller than for other types of DNA. So that's account for a unique origin. Some of you may remember reference 23 as one of the ones that we mentioned when discussing Y chromosome diversity of chimps and humans. Genetic data also indicate that all non-African populations are quite closely related. According to the out of Africa model, this is explained by a severe and very recent bottleneck of the order of 10,000 to 50,000 years ago that the non-African ancestors supposedly experienced. 10,000 years ago sounds kind of interesting, shall we say. This is perhaps the main reason why this model is more popular today than multi-regional evolution. African populations, on the other hand, look older, at least at first sight. There are also considerable genetic differences between African groups, indicating that their ancestors lived in small and relatively isolated tribes, which means, remember, less diversity within the tribe, but more diversity between tribes. Out of Africa model adherents interpret the bottleneck that the ancestors of all non-Africans supposedly experienced as descendants of a few thousand people that migrated out of Africa. For a long time, the non-African population stayed very small, but then they quickly expanded and diverged. These arguments for the out of Africa model seem convincing at first, but they could also be used for a unique origin model with an old African ancestry. The only difference is that the ancestral population of all people alive today, supposed to have lived in Africa before the migration and expansion to other continents took place, is a single couple with created diversity. But what about a founding couple from the Middle East? Is this unique origin version incompatible with the data? Not necessarily. This model requires that the age of humanity is much more recent. The reason that the originating pair replaces the long-lasting bottleneck that supposedly occurred after immigration to the Middle East from Africa, and the genetic diversity that remains after this bottleneck population is replaced by created diversity in the first pair. This would explain the relatively large genetic variability we find among humans today for non-sex and X chromosome DNA. Recall that the diversity of Y chromosomes is much smaller, and it is. A recent paper that compared sequencing among Y chromosomes found that a large group of Africans are in fact more related to non-Africans than to all other Africans. A possible explanation for this is several migrations into Africa from the Middle East. There are also tentative explanations within the Middle East unique origin framework for the observation that African populations look older than non-African ones for non-sex and sex chromosome DNA. Future research will tell how credible these explanations are. Block structure of DNA. A large part of our autosomal and X chromosomes have apparently been recombined into blocks of varying lengths. Many of them are on the order of 10,000 nucleotides long, but the variation in length is large. But even though the blocks are long, there is still very little variation within them. They come as blocks. Each block comes in just a few variants, for many parts of the genome. 4. Our chromosomes are different mosaics of these block variants. This DNA block structure is remarkably consistent with the unique origin hypothesis. If the first couple originated with DNA diversity, there would have been 4 different copies of each autosomal chromosome, 2 for each individual. Their 4 chromosomes have since been scrambled by ancestral recombinations, and today each of us has one mosaic of the 4 founder chromosomes inherited from our father, and another one from our mother. Inbreeding, depression, and genetic entropy. It is well known that many alleles will be lost due to genetic drift when a population experiences a severe bottleneck. The long-term consequence is decreased ability for long-term adjustment to environmental changes. But there is also a more acute risk of inbreeding depression when the frequency of recessive disorders increases, as more offspring receive from both of their parents a harmful variant of a disease-causing gene. Cystic fibrosis, sickle cell anemia, etc. The smaller the population is during the bottleneck, and the longer time it takes before its size starts to increase, the more severe are the consequences for the population's viability, so that ultimately it may die out. For humans, there are several well-known examples of the drastic effects of continued inbreeding, such as the extinction of the Spanish Habsburg dynasty around 1700. And the high occurrence of a severe form of color blindness on the Micronesian atoll of Pingalap after a typhoon hit the island in 1775, and 90% of the inhabitants died. Inbreeding depression is potentially a difficulty for the common descent model. Recent calculations reveal that the model predicts a very small bottleneck of the order of a few thousand individuals, and that it lasted for at least 1,000 generations. In spite of this, the survivors of the bottleneck are believed to have expanded and to conquer the rest of the world. It's a little odd that the inbred population seems to have done very, very well, raising the question of whether something is wrong with our theory here. A unique origin model avoids to some extent the problem of inbreeding depression if the created diversity of the first pair had only neutral variants, whereas the harmful ones appeared later through germline mutations. For a similar reason, a model with the young age of humanity has another advantage. It can handle the problems that the slightly deleterious mutations are so many that natural selection cannot remove them all. For an old population, as the mutations accumulate over time, the technical term for this is that the genetic entropy increases. You may remember the book Genetic Entropy. They have potentially very damaging effects. But if the pair lived recently, and if all present-day harmful variants arrived only through mutations in their descendants, there has not been enough time to accumulate them into large numbers. In contrast, any model with an old age of humanity faces a problem, since either the number of slightly deleterious or deleterious alleles increases, or a bottleneck occurred that removed some of these harmful variants, but at the cost of spreading others that could initiate inbreeding depression. Archaic populations, human or not. As mentioned in Section 5 of this chapter, significant fragments of Neanderthal and Denisovan DNA have been found among present-day humans, so that researchers suggested that some interbreeding took place between archaic populations and the ancient humans that supposedly immigrated out of Africa. This admixture is believed to have happened at least 50,000 years ago, and probably later on as well. It is in fact well known that gene flow between closely related populations is helpful in order to increase genetic variability and to avoid inbreeding, and indeed the archaic introgression is believed to have had positive effects, like helping the Tibetans to adapt to high altitudes and the non-Africans in general to adapt to colder temperature and to ward off infections. But the common descent model predicts a split between humans and archaic hominins more than 500,000 years ago. So how do they continue to interbreed then? It would therefore be remarkable if two populations after such a long time of separation were still able to have fertile offspring. But even if this were possible, because of the long separation, it is reasonable to believe that the offspring had low fitness, since our archaic ancestors had most likely accumulated many alleles which were deleterious for humans before the admixture took place. In view of this, it seems that the large fractions of archaic DNA among present-day humans is more reconcilable with a unique origin model in which Neanderthals and Denisovans are descendants of the originating pair and hence are our fully human relatives. Neanderthals are human. Denisovans are human. Maybe even Homo erectus is human. That means humans are around, if you believe the stuff, for 2 million years. We'll come back to that in my conclusion. Conclusions, we have argued that a unique origin model with either a younger or an old age of humanity with created diversity should have at least the same explanatory power for human genetic data as the most popular common descent scenario of today. Any model must be able to explain the big genetic differences between humans and other species, solve the problem of inbreeding depression, support the viability of human and archaic population admixtures, and give reasons why our DNA resembles a mosaic of about 4 founder genomes. The conclusion is that the unique origin model seems more plausible. Testing of the models. And this is the part of the chapter I like the best. The qualitative arguments of Section 5 lead us to conclude that the unique origin scenario of human history seems plausible. It is of interest to follow this up in a more formal way. The basic idea is to simulate genetic data from each proposed model many times and then compare how well the simulated output fits real data. This comparison should include nucleotide diversity, allele frequency spectra, LD plots, and other statistics. There are at least 2 different ways to proceed with these simulations. Forward simulation. The most straightforward way of simulating genetic data is to start at the founder generation and then proceed forward in time. For each simulation round, one first assigns genomes to the first pair of humans. Demographic and genetic data are then simulated one generation at a time using the heredity principles of Section 2. The main advantage of forward simulation is its great flexibility. Virtually any type of model for human history can be simulated and validated with real data. But the method requires the DNA of all humans is simulated. In view of the size of the worldwide human populations, this is computationally very demanding and requires long execution times. Backward simulation. There's another much faster simulation algorithm. A more detailed description of it can be found elsewhere. There's a reference. In each simulation round, only a small subset of genetic data is generated. The main idea is to first select a small sample of humans alive today, for instance, a few thousand individuals, then simulate their genealogy backward in time using a method called coalescence theory until the founder generation is reached. And presumably you could say what Adam and Eve's original genomes were. When the genealogy has been generated, DNA is assigned first to the founders, then forward in time to all their descendants along the branches of simulated genealogy. Genealogy will be only a small subset of the ancestral human population. We refer to this method as backward simulation since the genealogy is simulated backward in time. We are currently working on implementing a model based on backward simulation. The intent is to validate it with real data. This is a long-term project whose outcome we hope to publish elsewhere. Using this approach, our intent is to demonstrate that a unique origin model is able to replicate current human diversity as well as or better than the common descent model. Or perhaps to prove the opposite. Therefore, if more than one plausible account of human origins can explain the data, the common descent model of our origin from ape-like ancestors can no longer be claimed as conclusive proof that there could not have been a single first pair. Here we complete our one long argument for a unique origin of humanity begun in Chapter 13 with an outline of the importance of the question. In Chapters 14 through 16, we have shown that the fossil evidence shows an absence of intermediates between ape-like fossils and human ones, indicating that the story told by evolutionists of a well-documented continuous bridge between the two is false, or to be more precise, likely false. The genetic evidence shows that there are many more uniquely human elements in our genome than are commonly ascribed to it. significantly more than the oft-quoted 1% genetic difference between chimpanzees and us. Many of these elements come from what used to be called junk DNA, but are now thought to be functionally significant. There isn't enough time for all these differences to have been selected and fixed in the ancestral human population by purely natural means. And now for our chapter here, population genetic models say that we had to come from an original population of thousands rather than two, that those models are subject to question and can be tested against an alternative model starting with just two first parents. Taken together, our argument leads to the conclusion that it is unwarranted to discard traditional interpretations of our origin. And, of course, they're suggesting that theistic evolutions do not do so. Now, my take on all this, the chapter is the most ambitious attempt I've seen to try to explain human genetics with reference to only two parents. It also does not try to give premature conclusions, but rather to give a model and in ways of testing it, which is something I highly approve of. The data so far seems possible to fit into a model consistent with the biblical record, although more detailed study is necessary. I agree with him on that. One interesting question could be raised: if the best model includes a recent Adam and Eve, and Neanderthals and Homo erectus are human, is that compatible with radiometric dating? And if it is not, then do we question the genetic data or the radiometric dating? It is of interest that the mitochondrial mutations, when measured by actual historical genetic studies, are much faster than estimates based on evolutionary divergences. That's just a fact. Recombination in Africans seems to have happened significantly more often than in non-Africans. That is another fact referenced by one of their references. And the question could be raised, might it be possible that mutation rates in Africans exceed those in non-Africans for some reason? More cosmic rays, more, they're eating more mutagenic foods, who knows what. And if that is the case, then that would be a prediction that creationists of a certain type could make about the data and test that prediction. But that's my opinion. Now it's your turn. You'll comment over here, Jack Stout.
You probably covered this several times, but I'm not quite clear. If you look at the genetic diversity in humans and the genetic diversity in, say, chimps, is there enough of a difference to suggest a pair origin in one case and multiple origin in the other?
I don't know that that question has, the data has been looked at with that question in mind, and it might require looking at more data from chimpanzees. There are regional differences in chimpanzees. There's western chimpanzees and central chimpanzees, and eastern chimpanzees, and I think they have bonobos as well.
Well, chimpanzees was sort of pulled out of the air because it compared, but with almost any other mammal, for example.
Well, yeah, and that's an interesting question because you could also ask the same question about, clean versus unclean animals. If you're doing a Noah's Ark thing, there seems to be a difference in the initial population size.
Oh, I want to say the following carefully. It's not where I am, but I'm wondering how much complete violence would be done to our approach to this if we used an assumption of not just a pair of Adams and Eves, but that they're that's a restriction that isn't there for the rest of the animals.
Well, that's a good question. one can make interesting assumptions depending on how one views that particular part of the biblical record. If one assumes that the statements are meant to be universal, not only from the point of view of the authors, but from the point of view of God Himself, then I think you are stuck with everything going back to 4,000 years ago. If, on the other hand, one assumes that this is what they saw, but that there could have been a small island out, let's say in Australia somewhere, that didn't actually get flooded, and so there were what are sometimes known as refugia, then you might see a different pattern. And it would be interesting to test those two hypotheses against each other. Of course, you'd have to get more data. Now, one of the things that's absolutely fascinating is that from mitochondrial data, it appears that 90+% of all species, from ants to humans to ostriches, and interestingly enough, for some reason, including sea creatures, which one would normally assume didn't get into the ark and so didn't have a bottleneck at that point. They all go back to about 200,000 years by mitochondrial DNA. The people who reported this, at least one of them is reported to have said, I fought against this conclusion as hard as I could. Presumably meaning that at the end he had to surrender to the data. And that gets even more interesting when one realizes that the estimates for mitochondrial change are usually made on the basis of some kind of evolutionary hypothesis rather than on the basis of direct observations. And when you use the direct observations to calibrate the data, it comes out back down to somewhere in the neighborhood of 6,500 years, more or less. That's the center, and the range includes 4,000 years, easily.
One final question. Is there, I'm assuming, as a behaviorist, that multiple matings with different members of the opposite sex would be more frequent in non-human populations than human.
So that you wouldn't have pairing as much.
And that could affect the diversity substantially.
It would be interesting to look at that because, of course, it depends on the animal that you're talking about. There are animals where they are actually mated for life.
Yes.
And behave much like humans, in fact, more like humans than some humans do. On the other hand, there are ones that are totally promiscuous. And it would be interesting to ask the question, will that make any difference in how you model a population?
And I believe chimps fit this last category you mentioned rather well.
That is true. Both chimps and bonobos, as I understand. Yes. Oh, by the way, gorillas don't.
That's right.
Yeah.
I find it interesting that inadvertently they seem to be defending a biblical story instead of addressing the question of theistic evolution per se. This is not, well, it's very understandable to us and so on, but I wonder in terms of their basic premises if they aren't migrating a little away from that. The other point I would make is it seems to me that a good case can be made for multiple possibilities for organisms from a standpoint of survival. If you have different possibilities, you can survive better. And from humans, from a sociological aspect, what if all women look the same? Would this kind of be boring? Diversity seems maybe a desirable thing so that everybody doesn't look alike. But that's speculation.
Well, of course, what if all men look the same? That'd be boring too.
Yes, it would.
I won't get into that one.
Well, it suggests that if you're going to look at genetic diversity and you're not going to invoke miracles after the first one, then you're probably looking at absolutely designed humanity, designed for various features. And I agree with that. And the interesting thing of it is, it looks like those blocks, we're still able to find, 4-part blocks. I think there's one in HLA where there's a suggestion of 5, but 2 of those 5 are fairly closely related and could be, one could be a mutation of the other one. And you're allowed a certain amount of slop when you're trying to make a model of this kind, because mutations will change the block slightly. Go ahead.
Well, first, I want to congratulate you. You've done a masterful job of taking a tremendous amount of difficult data and simplifying it.
To be fair, the authors have. I'm just reporting what they say.
And I think you've done a very good job of arguing that there is an alternative model using the same basic assumptions.
The presentation is excellent.
Thank you.
That would perhaps coincide better with the biblical record than population genetics does. I'd like to point out, however, that there's also a third alternative model that's possible here that also coincides apparently with some of the biblical record. And to lay the groundwork for that, I would like to point out why the alternative model you suggest doesn't quite fit the biblical record as it stands if you take the Bible as being literally true. Because the Bible doesn't say that there was only an original pair. The Bible says that the son of the original pair went into the land of Nod and found a wife. Now, of course, the old explanation is she must have been his sister. But the Bible says that after Seth was born, daughters and sons were born to Adam and Eve. Why would it not mention an earlier sister if there was one? Then a little further on in Genesis, after Cain has murdered his brother, God's going to put a mark on him.
Take care of that, dude.
Yeah. And he says, they will kill me. And the Greek there, if you write it down, speaks of a tribe. So the Bible seems to suggest that there were a population of humans living apart from Adam and Eve at the same time that they existed. And if that's true, then they would be the result of an earlier creation. So the dichotomy is not between creation and evolution. You can have it both ways. You can have an evolution, and then you can have a special intervention in the creation at some much later point, which would fit your model. So we explore that in a book we've written called Space Age Interpretation of the Bible, but I just wanted to mention a third.
No, you're correct about that. The old Earth creationist people will not be too happy with that. And people who are looking at racial relationships may not be too happy with that either, because what it implies is that Neanderthals and Denisovans and Homo erectus of various shades are not actually human, and that some races of humans are kind of partly subhuman, apparently as much as 40% subhuman, mostly those paleoanthropologists, but
if God chose to create through evolution, he could transcend that.
yes, no, you're correct in that it raises interesting questions as to whether Neanderthals would be subject to the plan of salvation and so forth.
Yeah.
I'm not saying from that it's wrong. It's just that the wrestling with that question comes with the territory.
Good morning, Church. It's almost over.
Morning.
I always enjoy your take. 5 or 6 years ago, when I was 86, I am now in my 92nd year, and I asked a primer question. And I said, you will remember, I said, we were talking about the bottleneck. This is a primer question, really, to this subject, because our parents have always told us it was that Cain married his sister. I said, who was out there? Now, I am coming from Solo Scriptura. In our book, I'm sure you don't mind, Celestial Heirs, 40 years ago, heirs of the gods, we took care of this subject. You may not agree with us, but in 1978, in Heirs of the Gods, which has become a classic, and they call it a rare book at Amazon, this is the sequel to it. At Amazon now, Celestial Heirs. So my Bible tells me, and I think because in one of our quarterlies, our mother, Ellen White, speaks of symbolism. And I was raised on her milk almost. And I'm here with this book and with a warpath here, because I'm going to say something. And I love it. Solo Scriptura. When I go into the etymology, says in the Hebrew, call, the Hebrew word, and there are many more derivatives after that, but that one, when he says that the Lord God is going to put him out, and we've heard this from childhood, and Cain says, oh, now they come from the term when you're looking up tribes, but he says, everyone will kill me. Now, everyone, the word call in Hebrew, when I look at that now, we don't want to be simplistic and silly about it. I look up everyone, and then I look up land of Nod, and I go through all of that, and I am an ordained minister. I'm thinking people didn't know that. I don't flaunt it. But there was tribes out there. They were there. If you free associate do the hermeneutics and go to the archaeolinguistics, of which we are calling going to the root words, like you would do a dig in archaeology. Folks, this is the 21st century. we've got to get in our minds thinking about what is going to happen in terms of, and I'm not making a diversion, we can't bring this model from Methodism from the 19th century into this is all wonderful that Brother Dr. Paul has shown us, but we were recreated. And if you look, so Lee, take it, yes.
May I add one footnote?
Sure.
About bottlenecks? It is, of course, true that Adam and Eve would be a bottleneck as an original pair.
Right.
But the real bottleneck described in the Bible is not the creation of Adam and Eve. The real bottleneck is the flood, because the Bible says that all life was destroyed except for what Noah was able to take onto the ark. And the only humans on the ark were his family. We don't know exactly how large the family was, but that would greatly narrow the diversity that might have been inherited from Adam and Eve. So really, to deal with your model, it has to be dealt with not from Adam and Eve, but from Noah's family on the ark, it seems to me.
Yeah, I think it has to be dealt with in both cases. I agree with you. And I think there's... it would be interesting to see there's three authors, and the implications of what they're saying sounds like they come from three different perspectives, at least one of which is old Earth, old life on Earth, as I understand it, because of other statements she's made. At least one of those is old life, old Earth. So it would be interesting if you wanted to contact those people and see if some of them have theories that are kind of sort of related to what you have to say.
I want to hear what you were going to say.
Well, I was just going to say that when Christ said, "Have ye not heard that in the beginning," which sounds like beginning, he created male and female, sounds like a pretty significant bottleneck, if you want to use that term, at least, but that there wasn't much before that.
It does sound that way.
And then I would remind us, when you talk about Neanderthals and so on, their brain case was bigger than ours.
Yeah. Yeah.
They have rituals, ceremonies.
Yeah. No, it will be interesting to see what happens as they dig further and they look for more data and try to see what kinds of models can be fitted into it. And, it's interesting for us to start out making hypotheses. What's even more interesting is to see whether the data supports them. Mm-hmm. Come back next week and we will discuss the difficulties in the sociology of science, I think is the best way to put it.